Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we'll go over some practice questions regarding the biochemistry unit we just went over, covering amino acid structure, classification, acid-base properties, peptide bonds, and levels of protein structure. As always, please pause after the question and try to work it out yourself. And with that, let's start. Number 1. Which amino acid is not chiral? And this should be glycine. Recall that to be chiral, a carbon must be bound to four different groups, and glycine's side chain is a hydrogen, and it also has a hydrogen as another group, making it achiral. Number 2, which amino acid substitution causes sickle cell disease? This is, of course, the glutamic acid to valine substitution. Number 3, at physiological pH of 7.4, what is the net charge of glutamic acid? And this should be -1. Recall that glutamic acid has a carboxyl group for its side chain, so it has 2 carboxyl groups and 1 amino group in total. At physiological pH, both the carboxyl groups will have a charge of -1 each, and the amino group will have a charge of +1. This will lead to an overall charge of -1. Number 4. Which amino acid has a side chain pKa closest to physiological pH? Aspartate, lysine, histidine, or arginine? This is, of course, histidine. Arginine and lysine are very basic and have very high pKa's, and aspartate is acidic and has a low pKa, and histidine, as we know, has a pKa of 6.0, and that's why it's used in enzyme function. Number 5. Which classification best describes tryptophan? A. Polar uncharged, B. Aromatic, mildly polar, C. Acidic, and D. Nonpolar aliphatic. If you remember the mnemonic WIFI, where W, F, and Y are aromatics, you would know that tryptophan, phenylalanine, and tyrosine are aromatics. We also know that it is quite big with the double ring system, and it's mildly polar. So the answer would be B, aromatic, mildly polar. Number 6, what type of bond forms between two cysteine residues? This should be easy. It's the disulfide bond. This is when two cysteines oxidize to become cysteine, and the byproducts of this reaction are 2 protons and 2 electrons. Number 7, which of the following amino acids is most likely found on the interior of a folded protein? Aspartate, lysine, valine, glutamine. So to go through this question, first you should think what type of amino acid would be on the inside of a protein, and this would be a hydrophobic, nonpolar amino acid. So anything with a charge would be on the outside. And so we know that aspartate and glutamine are acidic and lysine is basic. And so these are all hydrophilic, and these would be polar and on the exterior, whereas valine just has an alkyl side chain and is nonpolar hydrophobic. So the answer would be valine. Number 8, what feature gives the peptide bond its rigidity? I hope you remember that it's the double bond character or the partial double bond character from resonance. This is due to the delocalizable pi electrons in the carbonyl and in the lone pair of the amino nitrogen. This exhibits resonance, giving the peptide bond in the amide partial double bond character. Number 9. What is the isoelectric point of a neutral amino acid like alanine? Is it 2, 5.5, 7.4, or 9.6? And the answer here is 5.5. And the isoelectric point formula is for neutral amino acids is the NH3 group plus the carboxyl group divided by 2. And we know the pKa of the carboxyl group is usually around 2, and the pKa of the amino group is usually around 9 to 10. And if we consider it as 9, then 9 plus 2 is 11. 11 divided by 2 is 5.5, giving us 5.5 as the answer. Number 10. Why are hydrophobic residues usually buried in the interior of proteins? A. To increase peptide bond strength. B. To reduce surface area. C. To prevent water from being ordered around nonpolar groups, increasing entropy and making folding spontaneous. Or D. Because they form strong ionic bonds with the other residues. The answer here is C. Recall that I said an increase in entropy means decrease in order, also known as increase in disorder. Systems move towards this, and this makes the folding process spontaneous. So C, to prevent water from being ordered around nonpolar groups, increasing entropy and making folding spontaneous is the answer. Number 11. Which of the following would most likely disrupt secondary structure without affecting the primary structure of a protein? A. Hydrolyzing peptide bonds. B. Heating to 120 degrees Celsius. C. Adding a reducing agent. And D. Mutating one amino acid. The answer here is B. Heating to 120 degrees Celsius. Let's go through this step by step. So by hydrolyzing peptide bonds, you'd be breaking apart the amino acids and therefore affecting the primary structure. A reducing agent would also break apart the cysteine bridges that would affect the primary sequence, and mutating an amino acid would also affect the primary structure. But heating it to 120 degrees Celsius would only cause denaturation, and this would not affect the primary structure. Number 12. A researcher replaces the leucine residue on the surface of a soluble protein with aspartate. What is the most likely outcome? A. The protein becomes more hydrophobic. B. The protein becomes more likely to aggregate. C. The protein becomes more stable in aqueous solution. Or D. The protein's secondary structure is eliminated. So aspartate is the deprotonated form of aspartic acid and is negatively charged. Leucine has a nonpolar, non-aromatic side chain. Leucine would not even want to be on the surface, whereas aspartate would want to be on the surface. Knowing this, the only option that really makes sense is that the protein would become more stable in the aqueous solution, option C. Number 13. Why is glycine considered a helix breaker in soluble proteins, but is often tolerated in transmembrane helices? A. It's hydrophobic and it stabilizes helices. B. It's too bulky to fit in helices. C. Its flexibility disrupts hydrogen bonding patterns. Or D. It forms covalent cross-links with proline. The answer here is C. Its flexibility disrupts hydrogen bonding patterns. 14. Which of the following amino acids is most likely to be protonated at physiological pH? Glutamate, histidine, lysine, or aspartate? The answer is lysine. Now this should be basic if you remember that when pH is less than pKa, groups tend to be protonated. You should know that the pH is 7.4 at physiological levels. You should also remember the pKa of charged amino acids. If you know all this, then all you would need to do is find a pKa that is higher than the pH, and if there's multiple, then you would need to find the one that's the highest. So right away, we can knock out glutamate and aspartate. From here, we just need to see which pKa is bigger, histidine or lysine, and we know that histidine's pKa is 6.0, and we know that lysine is basic. So we know lysine's pKa must be bigger, and from there we would just choose lysine. And if you remember the pKa's, then you would know that lysine is 10.5, and that must be the answer. So for this question, you don't even need to know the pKa's if you apply common knowledge. Number 15. Which amino acid would you expect to be enriched in the active site of an enzyme involved in acid-base catalysis? Leucine, glutamine, histidine, or valine? This is another easy one. It should be histidine. This would be found in the active site due to its imidazole side chain, which allows it to act as a general acid and a general base, facilitating proton transfer during catalysis. This shuttling of protons enhances enzyme efficiency and plays a crucial role in reactions such as acid-base catalysis. What are the 4 groups attached to an alpha carbon of a proteinogenic amino acid? The answer here is the amino group, carboxyl group, the R group, and hydrogen. You can remember this by corn, so CO is carboxyl, R is R group, and N is for the amino group, and you should just know that there's a hydrogen attached. Number 17. Are amino acids L or D, and are they R or S? Also, are there any exceptions? So you should know that amino acids are L. Glycine is not L because it's not chiral, so it cannot be L or D. And the 20 proteinogenic amino acids are R, except for cysteine. Since the sulfur groups takes priority, it is S. Number 18. Name the positively and negatively charged amino acids. This is just a memory question. You should know that positively charged amino acids are histidine, lysine, and arginine, and negatively charged amino acids are aspartic acid and glutamic acid. You can also use a mnemonic: dragons eat knights riding horses. That's D for aspartic acid, E for glutamic acid, K for lysine, R for arginine, and H for histidine. You know that 2 are acidic, and the ones that have acid in their name is probably a good bet for the ones that are acidic. Number 19, what charge would alanine have at pH of 1, 7, and 11? So you should note that it's an unneutral amino acid. So the carboxyl group has a pKa of 2, and the amino group has a pKa of 9. So at pH of 1, lower than the pKa, it would be protonated and have a positive charge. At pH of 7, it would have no charge, and at pH of 11, which is greater than the pKa of 9, it would be deprotonated and have a negative charge. Number 20, aspartic acid's pKa values are 1.88, 3.65, and 9.60 for pKa 1, 2, and 3 respectively. What is the pI? So for this, we should know that it's an acidic amino acid, and we use pKa 1 and 2, which is the carboxyl group and the side chain, and we would just average them, which would be 2.75. Number 21. For arginine, pKa 1 is 2.17, pKa 2 is 9.04, and pKa 3 is 12.48. What's the pI? You would use the same principle as before, but for amino acids that are basic, we use pKa 2 and 3 and then divide those by 2, and this would be 9.04 and 12.48, and so the pI would be 10.76. Number 22, for valine, pKa 1 is 2.32 and pKa 2 is 9.62. What's the pI? This should be simpler. As for neutral amino acids, the side chains don't have pKa's, so you just average the 2 that you were given and you would get 5.97. Number 23, what is released during formation of a peptide bond? This would be a molecule of water, as it is called a dehydration reaction. Number 24, where do trypsin and chymotrypsin cleave? So they both cleave at carboxyl ends. Trypsin cleaves to the carboxyl ends of arginine and lysine, whereas chymotrypsin cleaves to the carboxyl end of the aromatics, so phenylalanine, tyrosine, and tryptophan. Number 25. In a neutral solution, most amino acids exist as what? And this would be Zwitter ions. And the last question, number 26. Which of the following is a reason for conjugating proteins? A. To direct their delivery to a particular organelle. B. To direct their delivery to the cell membrane. C. To add a cofactor needed for their activity. Or D. All of the above. And the answer here is D. They have tags on them that indicate if they should be directed to the cell membrane or to a specific organelle, and they can also provide the activity for the protein. An example being the heme group is needed in hemoglobin to bind to oxygen. That's it for today's lesson and for this chapter in amino acids and protein folding. Next time we do biochemistry, we'll look at enzymes, but the next episode I post will be on biology and back to the cell.